Okay, so now we're going to talk a little bit about modern genetics, and we're also going to talk about how these are used throughout this entire course. So first of all, we could talk about it in terms of recombinant DNA technology. We have the ability to take any gene from any organism and put it into any other organism that we want. So for example, we can take the human gene for insulin, we can put it into bacteria, and those bacteria can produce insulin for us. And in fact, this does happen, and we use this produced insulin as a drug for many diabetics. We can produce many other things like human growth hormones and other genes as well. When we talk about cloning, we'll talk about this in a little bit more detail, but when we talk about DNA cloning, what we're talking about is making a copy of DNA. You can talk about cloning DNA, so we're talking about single genes or single pieces of genes, or you can talk about cloning organisms, in which we're not going to get into that a whole lot of detail, but basically you're making an identical twin of that particular organism. We'll talk about the Human Genome Project. This is a large project, and what it was to determine all of the sequence of the human genome. The result of this project has been identification of many different genes that are involved with diseases. And the benefits of this project are still ongoing, and we're learning much more about the human genome than we ever realized we could. Lastly, applications of genomics. So this is where, okay, we sequence the human genome, now so what? The goal of this is really to try to, again, identify genes that are involved with disease, identify genes that we can maybe produce in other organisms so that we can use these for potential drugs and so forth. Okay, so some of the most common model organisms are listed here. We use many of these because they are very cheap and inexpensive to use. It's also a lot more ethical to use, say, bacteria for research than it is to use, say, humans for research. So some of the most common ones, again, are listed here. So out of bacteria, many different types of bacteria are studied, but arguably E. coli is the most studied. Out of yeast, Saccharomyces cerevisiae is also very commonly studied. Roundworm, or C. elegans, is often used to study things like development or cell death. The fruit fly has been used for many different types of genetic experiments, as well as cell death experiments and other types of experiments, Drosophila melanogaster. The mouse is the most commonly used mammalian organism, and that's because it's very small, very easy to breed, and so forth. Out of plants, the most commonly used is something, it's basically a weed, Arabidopsis, and that's most commonly used for plants, although more of the economically, agriculturally important crops are now being studied as well, things like rice and corn and wheat. Zebrafish, also very commonly used in the lab. That's because they're very easy to work with. You just basically need a couple of different fish tanks that you can work with for these. Cell lines, also very commonly used in research. So one of the first things that people will do when they're studying many of these different things is using cells that have been grown from a human. The problem with using many of these cell lines is that normal cells don't want to grow and grow and grow for a very long time. These cells have been changed in some way. Many of them have arisen from tumors, and they are much more likely to be able to grow in perpetuity. You're allowed to freeze them down, you can thaw them back out, they'll grow just fine for you. Normal cells, not going to do real well with that. So the impact of biotechnology is really just beginning in the terms of genomics. Genomics is going to be the study of all of the genes of a particular organism. Proteomics is the study of all of the proteins of a particular organism. Bioinformatics is a really growing area of IT because we need really specialized programs and equipment and computers in order to understand and be able to analyze this vast amount of data that we're getting from all of the genomics, all of the proteomics.